confession, because I actually didn't know Alan before I invited him to give this talk, because we saw a paper published by him in Nature Biotech on using direct evolution for making MRI contrast for looking at dopamine. I think that was the paper. So then we read more about his work, and I thought maybe it would be interesting to invite him to give us some more details of his work. So Alan got his Ph.D. in 1998 from Harvard, and he has also been getting a lot of awards and other things before he is now at MIT. So one of the most notable awards, he got the Director's Award from NIH in 2007. So he's now an associate professor in biological engineering at MIT. He's also affiliated with the brain and pattern neuroscience, the nuclear engineering program, and also the Java Institute. So he's going to tell us about functional molecular in the brain. Thank you, Alan.
Well, thank you very much, Xiaoping, for inviting me. Thanks for all of you for coming here. And I can say that I've already enjoyed the warmth of Atlanta, both at a personal level and for those of you who know anything about what's happened in the Northeast this year, at a physical level. So it's a great pleasure to be here, and I'm going to tell you about my lab's efforts to perform functional molecular imaging in the brain. And let me begin with this. So one of the greatest scientific and engineering challenges of the 21st century is the challenge of reverse engineering the brain. And what's reverse engineering the brain? Well, it means building an understanding of brain function to the point where one could build machines that emulate brain function, possibly figure out interesting ways to repair brains, and obviously understand how brains work. And I probably hardly need to tell you that the applications of being able to reverse engineer the brain are multifold and range from artificial intelligence, design of neural prostheses, neurological treatments, and obviously my personal favorite, basic science, just understanding how the system works. So in some sense, reverse engineering the brain is the goal that all of neuroscience strives towards. So why is it particularly an engineering challenge as well as a basic science challenge? And I think this picture that shows some sort of egg-headed types working on what presumably is a dysfunctional brain, you see it seems to see a zebra as a chair, illustrates part of the problem. Notice what kinds of tools they're using to do this work. And I think you might imagine that using a set of wrenches on a brain, something that really performs unbelievably complex tasks, is really not the right thing to do. Now, of course, this is an analogy, but the point I want to get across here is that one of the key reasons why reverse engineering the brain is really an engineering challenge is because we need to find better tools to facilitate analysis of neural mechanisms. Just to illustrate the magnitude of the problem that we're up against, here's a brain. Brains have an extraordinary degree of complexity. So many of you know that human brains have on the order of 100 billion neurons. For even a rodent, it might be many millions. Each neuron has on the order of 1,000 synapses, connections to other neurons. And there are all sorts of different cell types that are defined by their patterns of gene expression, neurotransmitters, the chemical signaling molecules that they use, and of course highly complex anatomy and connectivity in a structure like this. So it's an unbelievably complex task. Yet, the gold standard technique for trying to understand brain function is based on tools that were developed decades ago. Electrophysiology, an approach where microelectrodes are buried into the brain and record the electrical impulses, which are thought to be the lingua franca of neural activity, but generally only from a relative handful of cells across the entire brain. So if you look at the fact that we have millions or billions of cells, usually we can only get these traces from at most a few hundred cells. So we have very specific information about electrical signaling, but very poor sampling and very little context about where in the brain and how cells we record are connected to the larger neural networks. So how can we get a more complete view of brain function? Well, we're very fortunate that there are techniques that allow us to measure brain activity at a whole brain level. And now the most popular sets of techniques are based on something called functional magnetic resonance imaging, which probably many of you know about in detail, or fMRI. fMRI allows us to obtain maps like the one shown here that illustrate the distribution of neural activity across brains. This is data from a rat that was getting an electrical stimulus to its forepaw. And you can see that there are parts of the brain in a region called somatosensory cortex and a secondary region over here that light up when the forepaw gets stimulated. And we can detect this using fMRI. Over here, this panel compares an electrical recording of neural activity in this region over here, shown in gray, so-called LFP, with the fMRI recording. This is from a contrast technique called BOLD. And you can see that the trajectory that fMRI measures is reasonably closely correlated with the electrical trace. So it is a measure of neural activity, and it's really a unique tool because it's non-invasive. It's based on MRI, this medical imaging technique that obviously owes its success to the fact that we don't have to slice up patients to get internal images. And we can see parts of the whole brain using this technique. But it's also a very limited tool for analysis of neural systems. Why limited? Because the contrast that gives us the ability to recognize brain activity in these images is based not directly on neural activity, but on the coupling between neurons and what they're doing and blood flow. And this panel over here illustrates a comparison between a set of neurons that corresponds to probably about 2 voxels, 2 pixels in this image here, and the corresponding network of blood vessels in a roughly comparable size. And what you can see is that there's this intricate network of neurons that are connected like a fine web, and we know that many of those connections are functionally important. But we can see if we look at the vasculature, the blood vessels in a comparable region, that the spacing in the blood vessels is obviously much more separated. This is a structure that responds with a point spread function on the order of a millimeter to changes that take place here. Why is it coupled at all? Because of the metabolic demands of the neurons and the chemical messengers that they release that in turn trigger a response in the vasculature. So we obviously lose spatial information as we go from neurons to vessels. We also lose temporal information. I think you can see that over here, that this fMRI trace is delayed and broadened with respect to the underlying electrical activity. What may be most important is that we lose information about specificity when we look at neural activity through the window of fMRI. And why is that? Well, here's an example that illustrates this. So imagine a hypothetical neural network that's made up of excitatory cells that use a particular neurotransmitter and inhibitory cells that use a different neurotransmitter, color-coded in red for inhibition and green for excitation here. Well, you could imagine that this neural network might be active with different balances of excitation versus inhibition, as indicated by these bar graphs over here. Yet the metabolic demands of those two situations would be quite comparable. So we might see a similar fMRI response for really radically different neuronal patterns of neuronal activity. And that's an illustration of how specificity, the loss of specificity in moving from neurons to fMRI keeps us from interpreting at a functional level what's going on. So how can we do better? Actually, let me skip this slide. How can we do better? I've shown you that we have on two ends of the neural recording spectrum electrophysiology, a very specific technique that provides very poor coverage. And on the other end of the spectrum, we have this fMRI, often based on contrast called BOLD, that gives us whole brain coverage but limited specificity and resolution. How can we merge the advantages of these two techniques? So my lab spends most of its time by trying to create molecular tools that are detectable by MRI, but that responds to neural activity at a molecular or cellular level. So the idea there is that we have a molecular level readout that includes the specificity of neural signaling, and that's combined with the MRI, the ability to image these things by MRI. So this is part of an overall kind of emerging field that's often called molecular imaging. We try to make molecular imaging agents that detect neural activity. And specifically, they're MRI contrast agents. What are MRI contrast agents? Well, most MRI contrast agents are paramagnetic chemicals. Here's a quintessential one over here. This is a molecule called gadolinium DTPA, which was one of the first FDA-approved MRI contrast agents. And most of them, at least the paramagnetic ones, exert their effects by interacting with water molecules. Many of you know that when you look at an MRI image, you're actually looking at water, the NMR signal of water molecules. And it's because contrast agents interact with water molecules that you get these effects. And in particular, the small magnetic perturbation due to the paramagnetic core of this molecule, in this case an atom of gadolinium, creates two types of effects on water molecules. It promotes something called T1 relaxation, which can be detected as a brightening in MRI scans that are T1 weighted. So more contrast agent, if it's a T1 agent, will brighten an MRI signal. And some contrast agents also create so-called T2 relaxation effects. Those are often referred to as T2 contrast agents. And they will tend to darken MRI signal as a function of their concentration. So if you imagine concentration going up here, a T1 agent will brighten, a T2 agent will darken. And you're going to see examples of both of these things in the rest of my talk. So that's why I'm taking the time to explain this. Now, the beautiful thing about MRI contrast agents is that they don't just do this in test tubes. So these are images of microtiter plates here. That's pretty easy. But if you introduce these things into living organisms, they produce similar contrast effects. So this is an example that follows the work of Alan Koretsky at the NIH, where a T1 contrast agent, manganese, has been injected into the eye of an unfortunate rat. And you can see that the eye became very bright. It's a T1 agent, so we're seeing the same effect that's illustrated here. Part of what's neat about this particular example is that the contrast agent is actually transported along fibers that connect to this eye. So here are two regions of the brain. This is called the thalamus, and this is called the superior colliculus, that are connected to the eye. The contrast agent moved to those structures, and so we can see them as hyper-intense or bright spots. Now, my lab is not just interested in tracing anatomy and in lighting up certain areas. We're actually interested in detecting neuronal function ultimately in real time. So we want things like this that brighten or darken dynamically as a function of what's going on in the brain in response to neuronal activity. So the challenges involved in this, of course, are designing, validating, and applying effective contrast agents for MRI studies of neural systems. And you're going to see, as my talk goes forward, that a lot of this has to do with designing appropriate chemicals that have the properties that we need. So how could we do this? Well, you can imagine that we could design MRI contrast agents that target a bunch of different aspects of neuronal function. Neurons do all kinds of stuff. They signal extracellularly. They release neurotransmitters, first and foremost. They're also ions in the extracellular space that change their concentrations. And we could detect all of those things. Intracellular signaling is a key aspect of neuronal function. Many of you know about neuronal plasticity and signaling pathways that involve calcium and kinase activity inside cells. Those could be handles on neuronal activity that we could sense with a contrast agent. And gene expression, of course, is also a potential target. And we've done work in all of these areas. But fortunately for you, I'm not going to talk about all of those things. I'm just going to talk about a couple of areas. So let me just summarize briefly the main topics that I want to cover in the rest of my talk. And I'm going to speak primarily about two things. The first is neurotransmitter sensing, with engineered proteins. This is the work that Xiaoping referred to in his kind introduction. And here I'm going to tell you about a new strategy for producing MRI contrast agents that's partly interesting for its chemical content, in a sense. I'm going to show you the first rudimentary MRI that we've been able to perform with a molecular sensor. And then I'm going to show you work that's now ongoing in my lab towards mapping behaviorally relevant patterns of neuroactivity with these sensors. And then I'll transition to the second topic, where I'm going to tell you just a little bit about some new genetically encoded MRI probes, protein-based MRI probes, for detecting intracellular signaling. Here I'll tell you about, in particular, protein contrast agents sensitive to calcium and kinase activity, which are key intracellular signals. And I'll show you some initial evidence that we have about calcium detection inside cells. And this is, some of you may not care about calcium, but it's actually very important. And in fact, optical imaging of neuronal activity is typically based on calcium signals inside cells. So it's actually a really important, potentially, and generalizable handle on neuronal activity. Okay? So let me start out with telling about neurotransmitter sensing. And so we had the idea that it might be good to be able to detect specific signatures of neural activity outside cells in the brain. Why outside cells? Because we don't have to think about delivering the contrast agent. Why neurotransmitters? Because cells in the brain often play functionally distinct roles that are dependent on the neurotransmitters that they use. So the neurotransmitter dopamine is a famous example. It is particularly important in some things you may know about. Reinforcement learning is an example. Some of you know about the so-called Skinner box, where you put a rat in a box and get it to do whatever you want in return for some kind of reinforcement, some kind of reward. And the neurotransmitter dopamine is key to the establishment and maintenance of those kinds of behaviors. Many of you also know about the role of dopamine in movement control, and in particular, these things are famous because of their dysfunctions. So a dysfunction of movement control is Parkinson's disease, which involves the death of dopamine-producing neurons in a part of the brain called the substantia nigra. Addiction is often thought of as a dysfunction of the reinforcement learning circuitry because of the chemicals, the addictants, the addictive substances that interfere with these neuronal pathways. So dopamine is important. We wondered whether we could try to understand the functioning of dopamine in neural networks by producing a contrast agent that could detect dopamine. And a student in my lab, Mikhail Shapiro, had the idea of trying to make a dopamine sensor based on a protein BM3 P450. So why work with this protein? Well, first of all, P450s, like hemoglobin, are heme proteins, and they're paramagnetic. So the heme, which is diagrammed in this brownish color here, makes this protein an MRI contrast agent because it's paramagnetic. Remember the slide I showed you a few back. There are other reasons to work with proteins, and one of the key ones is that we can engineer proteins with a speed and facility that at least my lab can't bring to the design of synthetic contrast agents. That was important in part because when you take an arbitrary protein like this, even a paramagnetic protein that's already an MRI contrast agent, well, it doesn't necessarily do what we want. In particular, for instance, this protein binds a ligand called arachidonic acid. It's a fatty acid that is very interesting in biology, but it's not a neurotransmitter, and it's not what we wanted to achieve. So we tried to engineer this protein to bind dopamine and to sense it by MRI. How did we do that? Well, we partnered with the lab of Francis Arnold at Caltech, who has designed high throughput ways to engineer these P450 proteins to do all kinds of stuff. Here we were engineering it to sense neurotransmitters, and it uses an approach called directed evolution. How does this work? Well, we take the gene for the protein, a piece of DNA, and we randomly mutate it. So we get a bunch of different copies of the DNA, each with a different mutation color-coded here. Then we take each one of these genes, put them in E. coli, and express them in multi-well plates. We lyse the cells, break them apart, and then assay them in the microtiter plates. We add successive amounts of dopamine or of the thing that we don't want these proteins to bind, arachidonic acid. And what we try to find are those mutants that bind dopamine better and bind arachidonic acid worse. This is a screen that's based on an optical measurement of affinity, but of course what we really wanted to detect were MRI changes. And so we then do a detailed characterization of the hits from this high throughput assay, the hits, the ones that showed the best properties, by MRI and optical measurements. And what makes this not just a screen, but actually evolution, directed evolution, is that we then take the best variants that we isolate at the end of this procedure and we feed them back into the cycle. So we go through successive cycles of mutagenesis, selection, and remutagenesis, kind of the way at least most people think animals evolve in nature, or even plants, right? Even bacteria. Okay, so this is directed evolution. And this is interesting in part because it's a new approach to generating MRI contrast agents. So people have used this kind of technique to modify protein functions over some period of time, but not for MRI contrast agents. How did it go? Well, the first thing we needed to tell was, is this protein an MRI contrast agent, and does ligand binding to it alter its properties as an MRI contrast agent? And we started out by measuring something called the relaxivity, which is kind of the strength of the contrast agent. It's sort of like quantum yield for a fluorophore. So we measured relaxivity of the protein, the wild-type starting protein, in the absence of ligand, in the presence of the natural ligand, arachidonic acid, or in the presence of very high concentrations of dopamine, far above anything physiological. And what you can see in both of these cases where we added ligands is that the relaxivity is depressed. So we did get a change in the relaxivity, and this is sort of abstract, but I think you can see the meaning over here. The higher the relaxivity, the brighter the signal in MRI, again, based on this T1 relaxation effect that I told you about, and in particular, these ligands both dim the signal here. So we can see that the protein, at least, is capable of functioning as an MRI sensor, initially for either arachidonic acid or huge amounts of dopamine. Could we make it better? It was key, actually, to our approach that the MRI signal change is accompanied by an optical signal change. So we got shifts either in the red direction, either in the blue direction or the red direction, depending on which ligand we added. And that was really useful because it enables us to assay the proteins at much lower concentration than we could do by MRI. And we could measure these titration curves. So what you can see here is that the binding affinity, measured optically now, for arachidonic acid is around a few micromolar, so 10 to the minus 6th in concentration. For dopamine, it's millimolar, around 10 to the minus 3. And why is this happening? Well, it's happening because we think these ligands, whether they be arachidonic acid or dopamine, are coming in near the heme, the paramagnetic part of the protein, and kicking out water molecules. So that's probably how this thing works. We now actually, I don't have this slide of it, but we have crystallography data now that backs this up. We've solved the structure of one of these proteins in complex with a ligand that shows that the water is being displaced. Okay, so this is how it works. Now, how did the evolution go? This slide shows the basic result, and what these are titration curves, again, now showing binding of dopamine, so dopamine is on the horizontal axis, to different variants, the variants that we produced after different numbers of cycles of that evolution process. And what you can see here is that over 4 or 5 cycles, reaching these color-coded yellow and purple mutants over here, we're able to achieve affinity down to about single digits of micromolar. So we started out with affinity on the order of 1 millimolar. We wound up with several hundred-fold improvement in affinity using this directed evolution approach. So it's a 300-fold improvement, which I was pretty impressed by when I saw it. And some of you may be wondering, well, okay, so it sticks to dopamine better, but what if it sticks to everything? Well, what this graph shows is that as we knock down the affinity, lower KD, for improve the affinity, lower KD for dopamine, we lost affinity to arachidonic acid. So this is over rounds of evolution here, and you can see that after about 3 rounds of evolution, we have negligible affinity for the natural ligand, arachidonic acid. So we do have some specificity for dopamine. Does it still function as an MRI contrast agent? All those data are about affinity. And what this graph shows here is, again, this relaxivity parameter, the strength of the contrast agent, in the absence on the left or the presence of dopamine. And this is for our best 2 mutants here. And again, you can see there's a contrast difference and a roughly 80% change in relaxivity, which is almost all or nothing. You can use these relaxivity changes to measure affinities, and the numbers come out pretty much right. You can ask, well, does it bind other neurotransmitters? So it doesn't bind arachidonic acid, but it may bind other neurotransmitters. And so we looked at a whole collection of neurotransmitters that might produce relaxation changes. And we found that most of them don't produce any effect. So for instance, some of the most famous neurotransmitters, glutamate, glycine, GABA, no effect at all with these contrast agents. Norepinephrine, which some of you know is chemically very, very similar to dopamine, did produce a response, but it was a weaker response. And the inset shows that the difference in affinity is about a factor of 4 between dopamine and norepinephrine. And for one of our mutants, serotonin, another famous neurotransmitter that we'll hear more about briefly later, produced an effect. So that mutant, the one color-coded in yellow that showed a response to serotonin, abbreviated 5-HT, that one is less specific, and as a result, we used the more specific variants for more of our in vivo tests. Nevertheless, these data show us that we really have here the first neurotransmitter sensors for MRI, and we thought about taking them and using them for initial experiments in vivo, even though, of course, we can make them even better. So what was the first experiment that we did? Well, it wasn't really in vivo. It was an in vitro experiment with cells. And what was involved here? Well, we have cells, a type of cell called PC12 cells that professionally secrete dopamine. And we could elicit dopamine release from these cells by hitting them with potassium, an ionic stimulus. If we give sodium, no dopamine release occurs, but it's a control because the ionic manipulation is quite comparable. We put our sensor, BM3H, or one of the variants of BM3, in the supernatant above these cells, and that's what's going to enable us to detect dopamine release in this test. Okay, how did this work? Well, here we're looking at relative signal, relative MRI signal in microtiter wells that had these conditions in them. And you can see here that the signal in the case of the potassium stimulus is lower than in the case of the sodium stimulus. And actually, I should have pointed this out on this slide over here. So note here that the relaxivity in the presence of dopamine, the strength of the contrast agent, goes down. The brightness goes down. So this result over here is exactly what we would predict based on binding of dopamine to the contrast agent. The signal should go down. Here it's a modest decrease. We can collect data in a way that enables us to measure an underlying parameter called R1, and that's changing also. In this case, because we knew the exact amount of contrast agent that we had, we could actually use these data to form a quantitative estimate of how much dopamine was present in this mixture. And what this graph over here is showing is the dopamine concentration in the supernatant detected by the contrast agent, either with the potassium stimulation or with the sodium control. And you can see that the concentration in the presence of potassium is somewhere in the 50s of micromolar, whereas it's around 20 micromolar in the case of sodium. So it's a reasonably big change. Now, is this right? This is what we calculate from our MRI data. Is it right? Well, we did a totally independent assay, something that doesn't involve MRI at all. It's based on an ELISA technique, a plate reader and an antibody, that actually came up with independent measurements of dopamine concentration. And I think you can see that they're not spot on, but they're pretty close. So in particular, in the potassium case, we're with an error. In the case of sodium, well, we see a pretty comparable difference between these two conditions. So I think we're able to do, what we can say from these data is that semi-quantitative detection of dopamine with these agents is possible, at least in vitro. Now, can we do anything in vivo? So this is a rat. And so we did a very crude experiment in vivo. We used the rat's brain as a test tube, but the rat was alive, so it's not really a test tube. We made 2 craniotomies. We drilled 2 holes in the rat's skull, and we injected cannulae to infuse in our contrast agent. And in one case, we infused the contrast agent only, on the right, color-coded in blue. And in the other case, we infused the contrast agent with our own dopamine. Now, why did we bother doing this? Well, one of our greatest fears with these things was that as soon as we injected them into a living environment, they would get chewed up and stop functioning at all. So we wanted to have a case where we knew that we had dopamine there, sufficient dopamine to produce a significant change in the binding of the contrast agent. And that's why we did this test. And let's see, is this going to work? No. Okay, I'm going to touch my computer here to play a little movie. What I'm going to show is a movie of the injection into the rat's brain over here. And over here on the left is the side that got the contrast agent plus dopamine. On the right is where the contrast agent is going on without dopamine. And I think all of you can see that there's this brightening here in the MRI scans as we infuse the contrast agent. I'm playing it again without dopamine. And the brightening is gone, basically, on the left. Why is that? It's because, remember, that dopamine turns off this contrast agent. It's a contrast agent that works in the absence of dopamine, but in the presence it gets turned off, and that's why it's dark on the left and bright on the right where there's no dopamine. So this was nice to see. We can quantify this basically by looking at the average signal over these time courses. You can see that there's a big signal change, about 25%, where we put the contrast agent in, but not when we put in dopamine with it. You can look at the time course here. This is the contrast agent plus and minus dopamine. The gray area here is where we were doing the infusion, then we stopped the infusion, and it's just sitting there. What about a control? Well, we actually have a great control for these experiments. Specifically, we have the wild-type protein that wasn't engineered to bind dopamine. One question you might ask is, could the protein be doing something to the system that interacts with dopamine in some non-specific way? Well, if we inject the wild-type protein, the one that doesn't bind dopamine specifically, then we could test for that. And I think what you can see here in these data is that we're seeing very comparable results in the presence or absence of dopamine when we infuse the wild-type protein. So what this shows is that we really are specific to dopamine. And these data, though a bit crude, are the first evidence of dopamine detection in vivo. Now, how can we detect a real process? So we cheated in that experiment because the dopamine came from us. We're not looking at a biological process. All we're looking at is what we're infusing. The good thing was that what we infused didn't go away immediately. It didn't seem to be disrupted by the living environment, but it's still not as good as detecting dopamine release in vivo and endogenous dopamine release in vivo. And so we designed a paradigm, so somewhat similar to what I just showed you, that tests for that. And what we're doing here is we're infusing the dopamine sensor, here called 8C8, on one side. We're infusing the wild-type protein, the control protein, on the other side. And with each of these proteins, we're giving pulses of high potassium stimulation. So remember that potassium was the stimulation that evoked dopamine release from our cells in our cell experiment. Well, dumping potassium into the brain evokes, essentially broad brain activity. And if you do this in an area of the brain where dopamine is naturally released as a neurotransmitter, then you'll get a lot of dopamine release. And this is well worked out in the literature. So this was a stimulation paradigm. And then, so we did this. And then what we did was more or less a sort of a conventional style functional MRI analysis where we correlate the signal trace as a function of time with the stimulus. And we found where in the brain was the MRI signal correlated to the stimulus. And here the stimulus again being these 3 pulses of potassium. And what you can see over here is that near the infusion point of our sensor, we found pixels that were correlated, whereas near the control infusion point, we didn't find that. This is from an individual animal. And we could take a group of animals and combine the analysis from all of those animals and actually map out the signal changes in response to these stimuli. These maps don't really mean anything because they're influenced by where the contrast agent spread from the tube tip. Each injection, as those of you know who've done infusions of things, each injection is a little bit different. So this isn't really a biologically relevant result, but what it shows is that at least a biological release of dopamine can be detected consistently by the sensor, and of course the control doesn't show this response. We could see little dips in the MRI signal over time, and if you look at the signal the map of signal over time during an injection cycle, you can see the signal change building up and then subsiding over the course of one injection cycle over here. So this is, again, a relatively crude experiment. Obviously, we're not learning new things about biology here. We've used this crude injection protocol, but it is the first time that we, or really anyone else, I think, has been able to demonstrate neural activity detection with an MRI-based sensor. So it's a key step in our overall plan to try to create a toolkit of these molecules that detect aspects of neuronal activity. What next? Well, these MRI sensors are not very powerful. So one of the things that one of the reasons why we had to do these injection procedures is because we need to get enough contrast agent into the brain to see it. So less invasive methods may not be good enough. Could we boost their efficacy as contrast agents? Well, that's equivalent to boosting their relaxivity, this R1 quantity. That's a measurement of the strength of the contrast agent. And we had the idea of trying to do that by increasing the spin, the electron spin, really increasing the essentially magnetic power, so to speak, of the molecule that's acting as a sensor. And my postdoc, Victor Lelyveld, student now postdoc, Victor Lelyveld, developed a pretty cool system for doing this. So we're producing all these proteins in bacteria. And it turns out that if you modify bacteria with something called with a porphyrin transporter called SHU-A, you can get them to take up whatever porphyrin analog you want. So porphyrin is the metal complex that lies in the heart of all of these proteins and makes them contrast agents. And so our idea was to fool these cells, basically to take up a manganese analog of what would normally be an iron porphyrin in the heart of the proteins. So we used the system to do this, and we were able to make fully folded functional proteins using the system. These are spectra. Maybe a few of you out there are chemists and you feel more at home with this than anything else that I've shown. What they show are that we can take essentially arbitrary metalloporphyrins and dump them into these cells, get them to be incorporated. These are, in the inset here, so-called CD spectra that show the folding of the protein and the perfect overlay of the blue and black traces here. Some of you probably can't even tell that they're two traces. That indicates that the proteins are well folded. So they are just like our sensors, but now they have manganese instead of iron. And this is the key point here, which is that if we now look at their T1 relaxivity, the same strength of the contrast agent business, in the presence or absence of ligand, so minus ligand in the solid colors and plus ligand in the pale colors, you can see that the manganese variant has about over twofold better response properties. So it's a stronger contrast agent, and it has a stronger response. And so this worked as an approach for reasonably seriously boosting the strength of this contrast agent. I think it's a gain that translates into our ability, for instance, to use a factor of 2 to 3-fold less contrast agent in vivo, or potentially to get a factor of 2 to 3-fold more signal in vivo for a specific experiment. Relaxivity improved by a factor of 2.5. All right. Now, we're impatient. So we want to actually start trying to learn things about biology using these tools. And what this figure shows here is a rat that is obsessively pressing this lever in return for rewards, in return for reinforcement. In this case, the reinforcement it's getting are electrical impulses through an electrode implanted in part of its brain called the MFB. It's a pleasure center. So the rat's actually having a pretty good time, even though it's embedded in an experiment here. You can see, right? Doesn't it look happy? Anyway, so we know that dopamine is really important for this process. Actually, this MFB reward paradigm is a very commonly used paradigm in studies of addiction. Could we map dopamine release patterns as a function of the stimulation paradigms in the behavioral context in this kind of system? So this is very preliminary results, and they're from my postdoc Taekwon Lee, and he started infusing the sensor into rats that are being stimulated in the way that this movie shows. And what you can see, at least in this example here, is that he's starting to see foci of what looks like activity associated with the infusion of the contrast agent and the stimulation here in a region of the brain called the nucleus accumbens, which is one of the main dopamine targets in the brain. If you look, now this is sort of choppy, but I think you can see that each one of these gray bars here is when we turn on the stimulus, what makes the rat press. And I think you can see that there are little dips in the signal that tend to be correlated with that. If you average over multiple cycles, you see a pretty decent trace of signal as a function of stimulation here. And so I think this is, quite preliminary data, but it's a step towards taking this new technology and trying to use it in a behaviorally significant context where we want to get, of course, two and ultimately three-dimensional maps of dopamine release patterns in the animal, ultimately, while it's performing the behavior. So this is towards dopamine release mapping in reward. We're also combining this with sort of more conventional Fmri studies, and Anna Fiallos, who just graduated from my lab, did some decent work sort of learning about the overall network that these things function in. So I think it's going to be an interesting direction. All right. So I've spent much of my time so far talking about dopamine. Dopamine neurons are very important. I illustrated that before, and we designed these MRI dopamine sensors to detect what these neurons are doing. Well, dopamine neurons project to large parts of the brain, but they're really only a small part of the story. Again, I flashed these figures up here. How can we see more? How can we see more about what's going on in the brain? How can we target other aspects of neuronal function that are represented by these different color codings here? Well, one obvious idea that some of you may have thought about is that we could use exactly the same platform to try to create sensors for different neurotransmitters. And we're doing that. So in particular here, we've developed, and this is again preliminary or pre-published data, developing a sensor for serotonin. It's another neurotransmitter, another monoamine that many of you know is, for instance, the target of SSRIs, Prozac and so on, related to Prozac. And what this figure shows here are relaxivity changes from a serotonin sensor. So we can see change that's quite comparable to what we observed with the dopamine sensor. These numbers here in the red box show the relative specificity, the relative binding affinity, for these different neurotransmitters, and you can see that it's very specific for serotonin. The KD is much lower. This is in micromolar, so it's submicromolar. And we're able to do these kinds of crude experiments in vivo with this. So far, obviously, we're moving towards more interesting in vivo experiments again. But a more general approach to looking at neuronal activity would involve targeting signals that all neurons share, and that therefore where we could apply the sensor to any part of the brain and detect neuronal signaling patterns. And so we have now a reasonably long-standing collaboration with a chemist, Steve Lippard at MIT, to develop small molecules that are contrast agents and that detect ions. They're based on porphyrins, and the idea behind that is that porphyrins, at least often, are cell permeable. So we can modify porphyrins. They're cell permeable, I should point out. They're also paramagnetic if you put in the right metal. This is something that you've seen already because they're in our proteins. So we modified them to be analyte specific, and in some data that we published pretty recently, we injected them into rat brains. We could show nice staining patterns in a region of the brain called the hippocampus. And then we did a subcellular fractionation of these brains to show where within cells our contrast agent is sitting. And we found that, in fact, most of it's in the cytosol. So it really is cell permeable. They're ion sensors. And right now what we're working on is trying to make a calcium sensor based on this platform. And this is really important, I think, because, again, calcium signaling is really the sort of gold standard for cellular neuroimaging. So most of the optical studies of neuronal activity use calcium-based agents, and this would be the MRI equivalent of it. We're far along in the synthesis, but we don't have it working yet. Instead, I'd like to spend the last really 5 minutes of my talk, or 10 minutes, talking about a different idea for trying to sense intracellular signaling, one that doesn't involve these small molecule or protein-based agents with one metal ion, but instead involves nanoparticles. And the work started in 2006 with a paper that we published. This is work of Tatiana Atanasievich in my lab. She made a magnetic nanoparticle-based MRI calcium sensor. Why did we work with nanoparticles? Well, some of you also work with magnetic nanoparticles, and one of the reasons why you do it is because they are known as the kind of atomic bombs of MRI contrast agents. They're particularly strong, particularly on a per particle level, as contrast inducers. So initially it was work of Ralph Weisslater and colleague that showed that if you cluster magnetic nanoparticles, their relaxivity changes, their potency as MRI contrast agents changes. They are T2 agents, so it's their so-called T2 relaxivity that's changing in these examples. What we did was we functionalized two populations of magnetic nanoparticles with two different proteins, a calcium-binding protein called calmodulin and a peptide called RS20 that binds calmodulin only in the presence but not in the absence of calcium. And the way I usually describe these things is you can think of them sort of like Velcro balls that get turned on by calcium but are off when there's no calcium, so they tend to clump when calcium is there. And what these two panels here show, these are so-called atomic force micrographs that show individual nanoparticles, here's the scale bar, individual nanoparticles in the absence of calcium, the presence of EDTA, and in the presence of calcium, you can see that these large aggregates form, okay? And you get a different mix depending on exactly what conditions you use. And obviously this wouldn't be interesting if there weren't an MRI response, and what this is showing is the T2, the underlying MRI parameter associated with these contrast agents, as a function of calcium concentration. What you can see is that as we add calcium, we move from dissociated to aggregated, and we increase the T2 in this system. And that translates into a pretty robust change in MRI contrast, and you can sort of titrate up or down the magnitude of the contrast effect depending on how you run the scan itself. Okay? So it was a pretty strong way to modulate MRI contrast based on these nanoparticles and dependent on calcium. But here's the catch. These are big nanoparticles. They're about 50 nanometers in diameter. How are you going to get them inside a cell where the calcium changes are taking place? Good sensitivity, but how to deliver to cells. So this is where we were inspired by the work of a couple of other labs on the protein ferritin. Ferritin is an iron storage protein of about 12 nanometers in diameter that accumulates iron oxide in sort of a biological analog to a magnetic nanoparticle that you might synthesize in your lab. The result is comparable in size to a synthetic nanoparticle, and it's got T2 relaxivity. And it also shows this clustering dependent effect that I just illustrated. In particular, if we cluster ferritins, now using a streptavidin biotin interaction, we can get changes in T2, here 1 over T2 is plotted, versus relative radius quite systematically based on the amount of clustering. Now, this is interesting for yet another reason, and this is part of why we worked on ferritin for this purpose, which is that a couple of labs, Eric Ahrens at Carnegie Mellon and Michael Neyman at the Weizmann Institute, had already shown that overexpression of ferritin in vivo actually can induce MRI contrast changes. So in other words, there are contrast changes that we could potentially modulate if we could do something like this, if we could modulate the clustering of these ferritin particles within cells dependent on the signal we were interested in. And so we started out with kind of a toy system where we modified ferritins genetically with protein domains that allow them to cluster based on a signal. And the first signal that we looked at was kinase activity. We were interested in that because it's closely related to calcium signaling in living systems. And it's an enzymatic process, so there are certain stoichiometric advantages. So we made a whole series of modified ferritins with different domains on them. And in particular, the blue and orange domains are the ones that really matter. The blue domain is something called the KID domain from a protein called Krebs, and that gets phosphorylated by kinases. When it does, it sticks to the orange domain, but not without phosphorylation. And so this is the picture of what we thought would be going on in this system. And when we tested it in vitro now, we were indeed able to see relaxivity changes. So this is R2, the measure of the strength of these ferritin-based contrast agents. And you can see that in these two yellowed conditions, with phosphorylation or the addition of enzyme, pre-phosphorylation or the addition of enzyme, the relaxivity goes up by about a factor of 2, and there's a corresponding image change that we could see in microtiter wells. Okay? We could use this to monitor enzyme activity over time, and you can see that the more enzyme you add, obviously the more change you see in a given amount of time. So this showed that this kind of effect, this clustering of ferritin for genetically modified ferritins, could actually induce MRI contrast. But obviously this is a long way from actually doing anything useful in vivo. In particular, we had a lot of constructs that went in to make these proteins. And getting all these things to express simultaneously in a given population of cells is not easy. We tried it. So we needed to move to a simpler system. And one of the key things that we tried to do was to split up the two interacting species between two protein platforms. So that I think one thing that you could imagine is if you try to express all those domains in one population of cells, you might not get orange or blue ferritins. You might get brown ferritins that have them all mixed up. Okay? So we tried to address that problem directly by fusing calmodulin, this calcium binding protein that I alluded to before, to one protein, ferritin, and we fused the interaction partner, RS20, to a different protein so that we couldn't get this sort of mixed formation of oligomers. So we wind up with two species, the red ferritins that have calmodulin, and these pink-green proteins that have the binding partner. And here the idea was that in analogy to our synthetic nanoparticles, the addition of calcium would promote the clustering. So calcium is here the trigger, not kinase activity. Sorry for baiting and switching. And we were expecting to see these clusters again. And so what this panel is showing is that actually initially our results were terrible. So using the wild type variants of these proteins, comparing minus to plus calcium, we saw nothing much. But under certain conditions, when we mutated this construct here, the pink construct, we saw a pretty robust contrast change between minus and plus calcium. It required getting the right balance of the two interacting species. And for those of you who like to think about these things, that won't be surprising to you. And in fact, it is a liability with the approach. On the other hand, we were able to see some evidence of responses inside cells. So we took these proteins, we stuck them inside a particular cell line, and we could see clustering because one of these interacting partners is fluorescent. It's kind of a neat trick that Victor, who did this work, built into the system. And I think what you can see here is that with the addition of calcium ionomycin, which is an ionophore that promotes calcium flux inside cells, you can see these little clusters of fluorescence building up here. So there's some evidence of a fluorescence response inside cells. Evidence of calcium responses. But if we look at the progression here, well, okay, we went from synthetic nanoparticles that are hard to deliver to these constructs that are just too complicated to implement inside cells. Now we've got this calcium-responding ferritin that is still quite dependent on the ratio of the two interacting species. So we still wanted something that was going to be more robust to signaling changes inside cells. And really the question we asked was, can we perform this kind of calcium sensing that I showed on the previous slide, but with a single protein? And this is current work in our lab. We have some preliminary results that are pretty encouraging in this respect. So we were able to make a single ferritin a single ferritin that's also fluorescent, stick it inside cells. Here we're using PC12 cells. This is a notation for the construct. And when we add this calcium ionomycin, again, you can see this nice punctate formation that probably reflects changes in the clustering and the organization of these constructs inside the cells. Of course, we've done appropriate controls. Here you can see a more blown up example over here. And possibly most encouraging, there seem to be MRI changes, at least that we can start to see when we take these cells, pellet them, and scan them. And right over here, what we're looking at is minus versus plus ionomycin. So again, adding calcium on the right, absence of calcium here. And we're looking at R2, this underlying parameter. You can see a little MRI change here and an R2 change. We've tried it with two different variants of our constructs, and we see qualitatively similar changes. So this is showing the degree of this decrease. And here we have a control protein that doesn't show very much change, although there's something going on there. All right, so this is really the first example of intracellular calcium detection with an MRI contrast agent. And I should emphasize that it is really particularly important that this is being done with genetically encoded contrast agents. And that's both because, of course, we can use the gene constructs to deliver the proteins by just expressing the DNA inside the cells, but also because we can potentially target different cell types with these constructs. So, supposing we want to sense dopamine activity, well, we have a different way to do it here. If we can just target the genetically encoded sensor to cells that use dopamine instead of all cells. And so in context of some other work, we're working with viral constructs that can selectively infect different types of cells. So this shows glutamatergic pyramidal cells, an important cell type in the cortex being stained. We can also use viruses. And so we can potentially ultimately perform cell-specific targeted functional imaging. Obviously, we still have a fair amount of work before then, and I don't want to gloss over that. We're also working on ways to boost the relaxivity of these ferritins, sort of an analogy to how we had to deal with that problem with our protein-based sensors. So let me sum up. I've told you about my lab's efforts to develop molecular imaging agents to sense neuronal activity. So MRI contrast agents that respond to neuronal activity with changes in their efficacy, and in turn with changes in the brightness or darkness of an MRI signal in the brain. And this is a sort of an approach that is pretty new. You can see that many of our results are rudimentary. Nevertheless, you can see that there is a lot to do, and we're trying to do it. It's an approach that we call molecular fMRI, and we're trying to get this off the ground. I focused my talk on a few different areas, and in particular, I focused on our first fMRI experiments with these metalloprotein-based dopamine sensors, the BM3 neurotransmitter sensors. I also told you just in these last few minutes about genetically encoded calcium sensors, and I touched on a couple of other topics. There are more projects that we have in the lab that I didn't have time to talk about. Obviously, the next steps involve doing a better job with delivery. You can see that all of our experiments so far have involved injections. We want to cover larger parts of the brain with these things and see more. And obviously, we have a lot more work to do to continue to improve and validate these MRI contrast agents, both in vitro and in vivo, ultimately leading to whole brain molecular level investigation of neural systems, which is really the long-range goal of all of this work. I'd like to thank the people who did the work. Let me call out some specific people. Tatiana did the calcium nanoparticle work. Let's see, going down here, Taekwon Lee did the work with the synthetic porphyrin sensors in vivo, and he's also involved in the in vivo, in the reward-related dopamine release work. Victor Lelyveld worked on the metallo-substituted protein-based sensors, and he also made these ferritin-based sensors for calcium. Mikhail Shapiro did the in vitro work with our protein-based dopamine sensors, and also our initial work with ferritin-based kinase sensors. And Gil Vestmeyer did the in vivo work with the dopamine sensors. We had some great collaborators. Frances Arnold has been our close collaborator. She's a protein engineer at Caltech. She was working closely with recently Eric Brustad and earlier Chris Odie and Phil Romero. I also alluded to our collaboration with Steve Lippard on synthetic contrast agents. We've had some other people that we worked with. I'd like to acknowledge funding from all these agents and smart molecules and all of you for your attention. So thanks very much. Happy to take questions.
We're a little bit over, but do you have some questions? Go ahead.
So you talked about the importance of calcium. For MRI purposes, we talked about manganese as a calcium analog. Has anybody tried, have you tried just injecting manganese to see if there's a dynamic functional change that can be detected?
Yes, so there's quite a lot of work with manganese as a contrast agent in vivo, and as you know, Alan Koretsky was the one who first explored that in detail, and I think the lesson from that work is that it does seem to be taken up in an activity-dependent way, but the dynamics are extremely slow and it doesn't come out on a useful timescale. So that's the answer. I mean, basically, if one is interested in performing Fmri on anything like the sort of behaviorally relevant neuronal timescale, manganese, I mean, it's better than nothing, but it's definitely not the desirable endpoint. Okay? So people have done good work with it. Yeah, sorry, go ahead.
With your extracellular contrast agents, it strikes me naively that you not only have a contrast agent, but also maybe a slight antagonist to the neurotransmitter itself. Do you have any thought about what the interplay between your contrast agents and the behaviors you're trying to look at?
Yeah, so you're absolutely right that a contrast agent, like essentially any chemical intervention, is going to disrupt the system. And in particular, the contrast agent will do something called buffering. It will soak up some of the neurotransmitter that we're trying to detect in much the same way that a pH buffer soaks up protons. So currently, obviously the hope in the long term, of course, is simply to use not to need very much of the contrast agent. Now, we're not at that point yet. We need a fair amount of the contrast agent, as I alluded to, so we will get buffering. And so there are two ways to deal with that, and one of them is to design experiments and look at phenomena where that's simply not going to be a big deal. So in particular, if we're interested in looking at what is the spatiotemporal profile of dopamine release due to a stimulus, well, that's largely a feed-forward question. It doesn't really depend on what dopamine is doing after it's been released. Okay? On the other hand, it's possibly less interesting from a behavioral standpoint. So method one is look at phenomena that are more accessible. Approach number two is, of course, to do all the behavioral tests in the presence of the agent, so that at least we have a good understanding of what it might be doing to the actual function of the neuronal network. So we do expect, of course, some interaction, and it will be important to do those tests, and we're set up to do them too. Any other? Yeah?
When you did the evolution,
Mm-hmm.
which method did you choose? Random mutagenesis, target mutagenesis,
Yeah.
or recombinant?
So for the work that I showed here, we used error-prone PCR. So I mean, this is basically the broader question for those of you who don't know about these methods is, essentially, did you just mutate every part of the protein and assume that each, or at least allow each one to have an effect on the properties of the protein? The answer is yes. In this case, we did. In some other work that we've done, we did mutate specific residues that we thought might be particularly important. Mm-hmm. Yeah, sure. Anyone else?
All right. Well, thank you again.
Thanks again. Yeah.